So what is blotting? The reason why blotting became popular in molecular biology is that it helps in probing of macromolecules, which is usually not possible in situ on the gel because of its fragile nature. And plus, it's incompatible with many of the probes and the buffers which is required for hybridization or antibody antigen interaction. So that is the reason we need a very robust, mechanically strong matrix on which you can transfer your macromolecules, be it DNA, RNA, or protein, and then you can probe the your specific sequence or your antigen using either immunological techniques or hybridization. So that is the reason why blotting became a very important tool in molecular biology. So as I've already told, there are three types of macromolecules which are transferred through blotting techniques. These are RNA, DNA, RNA, and protein. And they are transferred into a carrier so that they can be separated and often follow the use of gel electrophoresis. So what Dr. Naveen has told you is a step which precedes the blotting process. So first you have to use electrophoresis to separate your macromolecule, and then you use your blotting techniques to probe your specific molecule. So southern blot is a blotting technique in which you use to detect DNA. Northern is for RNA detection, and western is generally for protein detection. So the original blot was Southern because it was discovered by Professor Southern, and the rest of the names are actually just um, the fancies of scientists. It was Southern, so they started with Northern, and in the last slide I'll show you the whole compass of different names they have put. So the principle of blotting is the mixture of molecule is separated first. The molecules are immobilized on the matrix. The probe is added to the matrix to bind to the molecule, and this matrix can be the different membranes which are differently used for southern, northern, and western. So then you remove all the unbound probes because you put in excess. The probes are put in excess so that you have good chance of finding the target on your DNA, RNA, or proteins. Okay, so all of the unbound probe or excess probe is washed off to remove background, and then finally the probe where it attaches to your target that you can visualize, and this gives you the abundance of your target. So the history of blotting starts with 1963 when Hoare et al. First demonstrated that single-stranded DNA can be immobilized on nitrocellulose filters. So that was the first instance when scientists could actually take out them DNA and RNA molecules from the gel and immobilize it on a robust, mechanically strong matrix. In this case, was nitrocellulose filters. Then hybridization of nucleic acid was shown in 1965, and finally in 1975 when Edward Southern showed how the principle and the validity of the blotting techniques for RNA, it was known as Southern blotting, and then after that different blotting techniques followed later on. So it's quite an old technique. Okay. So now, can you see the whole slide, or is it cutting its cropping from the edges like it was before with Dr. Naveen?
So we can
So
see the
just
whole
let
slide.
you can see the okay, then it's fine. Then I'll use this slide view. Okay, so southern first go for the southern blotting because it was the first blotting technique. So this involves separation which is generally done using axial electrophoresis, and transfer and hybridization. And this DNA can be a single gene or a part of a piece of a DNA viral genome. And the key of this method is hybridization. So you take your genomic DNA and then you first chop it off into small fragments, digest it so that it's easier for the probes to bind and for recognition. So this is how it all goes. So you run your nucleic acid, and in using electrophoresis, and then you use paper towels to make a sandwich. So you put gel, your agarose gel here, and your membrane here, nitrocellulose membrane, and you put filter papers, and then over that you generally put a weight. So the separation, mostly people use capillary action for transferring DNA and RNA to the matrix, or which is called the membrane. For proteins, the different type of transfer method, which is called electroblotting, is used, which is much faster and which is much needed for heavier macromolecules like protein. So once you have the transfer of your DNA to your membrane, nitrocellulose membrane, then you use your probe, which in previously used people used to use radioactive probes, which was P32 label nucleic acid probes. Now with the discouragement of using radioactivity, people are also using fluorometric probes to detect their target sequence. So once you have sealed this bag containing your membrane and your probe, then you do some washing steps, and finally you use autoradiogram if it's a radioactive probe, or you use a chemidoc if it's a chemiluminescent probe, or detection using a compatible instrument which can detect fluorescence. So that is how southern blotting is done. So matrix I have shown is actually creates a capillary action. So the nucleic acid is transferred from the gel to your membrane using you apply weight, and then the capillary action helps in transferring it. So what are the applications of southern blotting? So southern blotting initially was used to do gene discoveries, mapping. It was used in evolutionary biology to see the evolutions of different DNA sequences in organisms. It's also used for diagnostics and forensic uses in still uses, although it's not very much used now, but still there are some cases in which these are used for diagnostics. So next, macromolecules which is transferred using blotting is RNA, and the blotting technique is known as northern blotting. And this is the process of northern blotting is very much similar to southern blotting. The only thing is in southern blotting we use the membrane which is nitrocellulose. For northern blotting, nylon membrane is used because of the compatibility with RNA, and the detection follows the same principle. Either it is a radioactive based detection of your specific RNA molecule, or through chemi or fluorescent detection. Now, one of the most important use of northern blotting before the real-time PCR was a trend was to detect to see your relative gene expression. So suppose you have added your drug to your cells, or injected in animals, and you want to see whether it affects the transcription of your target gene, then you generally use northern blotting to do that. And beside seeing the relative gene expression changes, you can also use it to see specialized changes in the RNA molecules, like RNA splicing, RNA degradation, or a change in RNA half-life. These are very much possible using northern blotting. But to do a northern blotting is quite labor-intensive work, because you have during all the process there is always a risk of RNase contamination, and that really affects the experiment. So so that was one of the reason why when real-time PCR became popular, people stopped doing northern, and hardly anybody now do northern blotting. So in nowadays, if you see papers, you don't see northern blotting at all if people have to show a relative gene expression. So one of the techniques which is still going on and probably will go on, it's almost a 30-year-old technique. It is not becoming any automated, but still it is much used in all of the basic science paper you see nowadays in big journals. And in all the journals, if you have to see relative expression changes or your knockdown efficiency, anything you have to do, you have to use this technique known as Western blotting. So unlike its other cousins, southern and northern, this is still a happening technique. So Western blotting is basically follows after you do your vertical electrophoresis or the SDS page assay. So as Dr. Naveen has already introduced you with the page, I'll give you a brief account of it. So what you do in SDS page is that you take your sample, which can be a tissue sample, cells, even bacteria, and what you do is you first homogenize your samples. So you use buffers. One of the most popular buffers is called the RIPA buffer. So you just burst open all the cellular compartment if you want a wholesale lysate. Otherwise, you can also use different buffers to isolate nuclei, mitochondria, or different subcellular organelles. And then you use your buffer to separate your buffer to make proteins from these specific compartment. So suppose you want to assay expression of a gene in a whole cell lysate. So you add a RIPA buffer. It has SDS and Triton X, these things, and then it opens the membrane of the cells, and you have a cocktails of protein floating in your solution. Now what you do is that you use a lamelli buffer, which has the bromophenol blue, glycerol, and SDS, and you mix it with your protein with your sample tissue lysate or cell lysate, and you run it on a SDS page gel. So you load the gel, and then you start your power supply. The proteins will separate because of their molecular weights, because charge being same on all of them because you're using SDS. So once the separation on the molecular weight has been done, then what you do is these are all the steps which are needed. So you are here. So you have already separated the protein on the gel. Then you do a sandwich of a Western blot. So it's similar to what we do with the Southern blot. Only the orientations are a little different, and it's not based on capillary action of transfer. You use electrical charge to transfer. So protein being negatively charged now, they are applied against the current in which they bind to the protein to the membrane, which is towards the positive. So the negatively charged protein bind to the positively charged membranes. Okay. So then after you have done this, I'll go one by one on all the steps. Briefly, after you have transferred, you check whether your protein is there on your membrane. Then you do all the washing you add your primary antibody, which is a probe in Western blot, and then you wash to remove all the unattached probes or antibodies. And then you use your secondary antibody, or if it is a direct antibody based detection, you just proceed for exposing it on a film or a machine which can detect chemiluminescence or a fluorescent uh, signal. So now going step by step in all these techniques. So as I have told you, unlike the southern and northern blotting techniques, it's not based on capillary action. So we use what you call as electrotransfer. So there are different types of ways in which you do electrotransfer. The old method and still very important way to do electrotransfer is through using a wet transfer system. Wet transfer system is that you submerged your so if you can see here, these are the cassettes. So they have these sponges, and then you use filter paper, like Dr. Naveen told you about Wattman paper. You place your gel and your membrane on both sides with the filter papers so that they are kept moist and helps in the conductivity of the buffer. And then you seal the whole cassette and put in a transfer assembly, which has transfer buffer, and it's totally immersed in it. So it's a lot of transfer buffer used to do that, and it is because of electrical conductance. The charge negatively charged protein will slowly leave the gel and go and bind to your membrane. So this is a wet transfer. It is very good for if you want to transfer your heavy proteins like more than 80 kilodalton. But it takes a lot of volume of transfer buffer, and it takes a lot of time, and you need regular cooling of the transfer buffer because it really gets hot. The other newer method of transferring proteins are semi-dry methods in dry. So most popular is the semi-dry method, in which you use a transfer assembly like this shown here. So the distance of the membrane and the gel is very much reduced in this type of system, and you apply very high current to transfer in a very short time. And the transfer buffer is also used in a very low amount. So it's generally good for fast transfer. So suppose you are taking 3 hours in a wet transfer, you will do it within half an hour. And even if you use some of the so-called specialized buffer by the companies, you can even transfer your protein in less than 5 minutes. So anybody, if students do want to do Western blotting, our lab is well equipped to do both wet and semi-dry. So if you want a demonstration, you can come here, have a hands-on, and see the different methods of electrotransfer. And dry is basically a very specialized instrument developed by Thermo. So I'm not going into details because they say the transfer is in less than 2 or 3 minutes. But the consumables are patented, so they are quite expensive. You can only buy it from companies like Thermo. So these are the two most of the time you will be using, wet transfer or the semi-dry method. So again, this is for semi-dry, you have the filter paper, then the gel, then the membrane, and then again the paper. For wet transfer, you need sponges also because you need a lot of conductance through the buffer. So the sponges are there and paper, gel, membrane, paper, and sponge. So that's the sponges are added. That's the only thing which is changed. And then again, you see the protein will go towards the positive anode, and that is how your membrane will absorb all the protein molecules. Now, there are two different types of membranes which are used for Western blotting. So, wait a second. Yeah, so one is the nitrocellulose membrane and the other is the PBDF membrane. So it depends on, we use regularly both of these membrane. It depends what is our interest. So suppose, for example, you want to reprobe your protein. So you have a gene of interest X, and you don't want to run a new gel to detect another protein which is Y, and you know they are separated quite fairly in the molecular weight. So you can use the same membrane to first probe your protein A. Then there is a step known as stripping of the membrane, and which allows you to get rid of the previous antibodies which are bound to your first protein. And then you can easily go on to detect your second protein of interest, which is Y, and later on even the housekeeping gene by stripping process. Now this is only possible in a membrane which has a very good adsorption and binding capacity for your protein. And this is only possible with PBDF membranes. So PBDF membranes are generally considered better for Western blotting because they have a high binding capacity. So you can reprobe the membrane at least 4 to 5 times if you wish. And they're also good if you want to send your protein samples on a membrane for mass spec proteomics. So you cut out that part just like you cut out your gel for DNA for sequencing. For membrane also you cut out that area of interest and you can send it for mass spec. But this can be only done if you're using a transfer with PVDF. Nitrocellulose is good. It's much more economical. It's cheaper than PVDF. It's high affinity for protein, therefore high retention. It has a comparably high affinity, but definitely lower binding. So it binds very easily, but it loses them also very easily. So it takes time for you to get your protein bound to PDF. It takes lesser time to get it on nitrocellulose, but the same thing happens for losing also. The nitrocellulose will bind it fast and also lose it fast. So you cannot do continuous stripping on a nitrocellulose membrane. And they are also very brittle, so they just break very fast. So they are a little bit difficult to handle a lot. Now, this is very important. So when you transfer your proteins to your membrane, and before you want to go for detection or of your target, you should always ensure whether the transfer is good or not. So most of the time, what happens that when you place your sandwich, you put your gel and the membrane, sometimes water bubbles come in the between. So you have a roller to get rid of those bubbles, but sometimes it's not perfect, especially on your new students, you will be doing hit and trial, and we also had the same problem. Still, we have the same problem. There are a lot of bubbles sometime coming. So before we go and proceed for detection, you have to understand these, and the antibodies are quite expensive consumables. So you don't want to waste antibodies on a membrane which doesn't have a good transfer. So, which is very essential. Then after transfer, you use this technique known as Poncho staining. There are other stains also, but this is the most popular one. So, unlike the Comasi stain which you do in your gel, this stain is a reversible binding to your protein on a membrane. So it will when it will bind to your protein on the membrane, and you wash it with water, you see quite nicely the bands are coming. But if you do a regular rinsing with water, you will get rid of all of these stains very fast within a few seconds. So it's quite good. You can see your protein integrity. First of all, your protein is not degraded. The transfer efficiency is good. Your molecular weights, if you suppose you want to transfer a high molecular weight protein, then you have to be sure that your molecular weights have been transferred. If the protein is around 100 kilodalton and only markers less than 50 kilodalton shows up in the transfer membrane, then you better don't proceed with the blotting, because your protein of interest also would have not been transferred to your membrane. So this is very important before you go for your antibody incubation. Just confirm your protein has been transferred well or not. Then after you're done with the ponchuous and seen the integrity of your protein and the transfer efficiency is good, then there's a process known as blocking. This is also done in other blotting techniques, and it's also very important in Western blotting techniques. This is important because the membrane, by its virtue, is binds or absorb loosely other proteins also. So for example, your antibody is also a protein. So your antibody may be non-specifically get bind to your membrane, which is free. So some of the membrane is being filled up by your protein, which has been transferred from the gel, but there are still some of the spaces which are free. Now, suppose you add your primary antibody and a secondary antibody, and these free spaces bind your primary antibody, then these will give non-specific signal or background. So we want to saturate your membrane as much as possible. So you add what is known as blocking reagents. So this can be non-fat milk, which is one of the most popular because it's one of the cheapest. Then people also use bovine serum albumin. So these are just non-specific proteins, which just fill up all the empty spaces in your membrane, so that there is less chances of non-specific binding of your antibody. So this is how it works. This is your target protein, which you have transferred through your gel. And these are all the free spaces in your membrane. Okay. And this can be filled up by, specific proteins in your blocking buffer, which contain milk or BSA. So once the protein is always is there, your membrane won't bind other proteins like antibodies. So you won't get non-specific signals. So this is very important. Never proceed for your primary antibody incubation before blocking your membrane. Now, after you're done, then the very important, then the main step is to add your primary and your secondary. So there are many ways in which you can detect your target protein. First is just use a primary antibody, which is tagged with a detection chemistry. So detection chemistry could be a color-based reaction, or could be a light-based reaction, which is called chemiluminescence, or could be a fluorescence-based reaction. So once your primary is bound, you can attach your chemistry here, which could be an enzyme or another things, and then you can detect your target on the membrane. Okay, this eliminates the chances of usage of secondary antibodies, which are not actually that expensive, but still can give you background if they bind non-specifically to other proteins. But then the drawback with this is that your signal amplification is reduced. So you have one antibody for one antigen, and then you have one enzyme per antigen. Whereas if you use a secondary antibody, generally it's not like what you see in this figure. Is actually many secondaries can bind to one primary. So you really have a amplification of your signal. So for each antigen, you can have at least 3 or 4 enzymes there to give you signal. So it increases your signal. but at the same time it may increase your background also. So that is a trade-off you have to understand. But still, most of the people still use the indirect detection. So you add your primary and then you add your secondary antibody to detect. Now, these are the different chemistry. I told you that it could be a colorimetric detection, which means that you add your substrate and it will turn out blue, like if using alkaline phosphatase enzyme here. You can see on the membrane itself that how it is showing up. Where is your protein of interest showing off? And one of the most popular detection chemistry still in use is the chemiluminescence base. So it is much more sensitive than your color base, and also there's you can use your film, X-ray film, to detect it. So it increases sensitivity, and also the linear range of detection is much more better than using a and one of the newer method is fluorescence-based detection. So this is still quite a new method for detecting target proteins using fluorescent base. So you have a fluorescent dye here, which will fluoresce. The only good thing about fluorescence-based detection is that you can do multiplexing on the membrane without stripping. So suppose you want to detect A, B, C, three proteins there on your membrane, and they are fairly well separated in terms of their molecular weight. You can add antibody against protein A, which will give a green fluorescence like CF488, and the B protein you can use antibody which is a different color fluorescent, and third is so in one go using specialized instrument like this called the Lycor, which this one of the instrument, even Biodad MP are some of the chemidocs which can do that. So in one go you can assess the expression of three or more genes without stripping the blood. Whereas in the chemiluminescence based detection, you have to detect first because the color is the same. So you cannot do multiplexing easily. So you have to use first, you have to do your develop your first plot, get your signal, then strip and then develop your second plot. So these are the different chemistries which are employed to detect Western blotting. Now, Western blotting inherently is not a quantitative technique, it's a semi-quantitative technique because what you get is the relative changes. You cannot get exact the amount what is there. So it's not like real-time PCR, which is a quantitative technique. Western blotting still is a semi-quantitative technique, but it can be made quantitative in some terms when you use softwares which can do what you call as densitometry. So you get a numerical value of the density, pixel density of your plots. So for example, this is your control. Suppose this was your control lane, this was your treated lane. Okay, so now your specific gene of protein of interest is detected more in your treated. So definitely you will say my expression of my target gene protein is more in my treated samples. But how will you make a graph and do statistical tools to use on that? So for that you need some numerical value. So for that we use softwares which are densitometric softwares, and they measure the relative density of the pixels. So pixels, when you see that black band coming, it the more the expression, the density of the pixels plus the area they take will be more. So it quantitates that pixel density and gives you a integrated density of your band, and that is what you plot on your histogram, and you see people showing histogram and target the protein is more and this is low. So this is how you quantitate a Western blot. So you isolate, you validate antibody and use your, but definitely you have to use a loading control so that any error while you were pipetting the pipetting the samples could be normalized with your housekeeping. Now, what are the housekeeping genes or proteins you have to see? So it depends on your work. So generally there are three main housekeeping proteins we use. One is GAPDH, actin, and tubulin. But as sometimes there are some drugs which can even alter the expressions of housekeeping genes. So you have to best if you can do at least two housekeeping for a protein just to see whether your housekeeping is not affected. And then when you're sure that a housekeeping is truly a housekeeping, then you normalize the pixel density of your target protein with that of your housekeeping. So ideally, your housekeeping should not change if with your treatment, and unless your pipetting error is there, they all should be almost same in all of the well. But if you have a pipetting error, the normalization with housekeeping can take care of it. So that is how you do a housekeeping control. And then there are some tools also to see whether you're not overexposed. So suppose you are exposing your blot a lot, then it won't give you a true picture of the linear increase in expression or decrease in expression, because after some time, even if your protein concentration is more, the signal cannot go more than that. So you have to be in a linear range of absorbance. Okay, so then you use your tools like Excel or GraphPad to plot the histogram, and then you use statistical power to say whether these differences were statistically significant or not. Okay, so that is how you make your electrophoresis and blotting technique a publication quality figure. So that you have to quantitate your blots. It's not like putting your blot alone. You have to quantitate it. So this is example which I'm taking from one of my paper we just published last year. So we were trying to understand the relationship between lysosomes and peroxisomes. So, you know, these are one of the most important organelles in our cells. So it was they were discovered in 1970s, but still it was not very clear how these organelles crosstalk with each other. So what we were interested was to see whether if you have a lysosomal defect, because in most of the metabolic disease or and in neurological diseases, you have lysosomal disorders, and these affect peroxisome. So if people, some of you don't know what peroxisome do, is that they are helpful in burning fats. So like your mitochondria is used to burn fat, peroxisome is also used to burn fats. So they are a hub for fatty acid oxidation. So what we were able to show using Western blot, so what did was that we use an inhibitor of lysosome, which is a baflomycin A, and we use fluorescent microscope to show that when you use a lysosomal inhibitor, then a lysosomal staining. So this is the stain which is a lysotracker stain. It goes down in your lysosome. But then again, to validate it, to confirm it, we went to Western blotting techniques. So what we did is that we detected that activation of a protein which is there in the lysosome. So lysosome has an acidic environment inside. So the proteins inside the lysosomes are only activated then when they have an acidic environment. So if your lysosomal inhibitor is working well, then you should not see an activated protein here. Of this is a cathepsin protein which is activated only in an acidic environment of lysosome. So you can see when you use a lysosomal inhibitor, you don't see cathepsin activation. And then again, as I told you, how do you quantitate? Then you do use softwares like ImageJ. This is one of the freely available software. You can quantitate the density of this band and the density of the band here, which was almost gone. So then you do it in triplicates or as many as time you want, because you have to apply it statistics. So you have to have a triplicate, at least triplicate, and then you make a histogram, and then you do statistics. So that is how a quantitative Western looks like. Okay, and this gives you an inference that indeed when you use this inhibitor of lysosomes, you have a defective lysosomal activity. And then again, when you have an inhibitor of lysosome, you don't see expression of peroxisomal genes. They go down. So they are reduced, significantly reduced. So this is how we use, this is just to show you how we use Western blotting in papers. So this is an example. And beside this, in your research work, when you see the expression of the genes, you can also use it to assess the mass of a splice variant of your protein also. But that you have to first load a known quantity of your protein, and then just like you do for protein quantitation, you can assess the molecular weight of the protein, unknown protein also, like a spliced version of your protein. And in diagnostic, it's the confirmatory test for HIV screening and also prion disease and mad cow disease. So generally they are not present. So it's yes or no type of thing. So it's not relative. It's just either the patient has a anti-HIV antibody when you do a Western blot, or he doesn't have it. And the same thing goes for the prion disease, and also a disease known as Lyme disease, in which they employ Western blotting to see protein in the disease condition. So now, as I've told you, there is an array of blotting compass. So first started with southern, now you have almost in all directions it's going. So these are the three which I told you. One is the now even I cannot find out. So okay, western is here, northern is here, southern is here. So these directions, the other directions were left. Then this people just use these terms, but actually they are used for different. Like if you want to detect a post-translation modification, there's a type of blotting which is called Eastern. Okay, now there's even a Middle Eastern there for RNA, DNA, and then lipid post-translation modification. You have a Far Eastern blotting. Then you also have for DNA protein interaction, Southwestern, and electromobility shift assays. So these are different arrays of blotting methods which can be employed to detect different changes in macromolecule as alone, or also in combination with other macromolecule. So with this, I want to end the talk, and I hope this will be useful for you if you're planning to do some protein work. And if you need any help from our lab, because we are doing Western almost daily, so we can help you with that. And best of luck with your doctoral studies. Thank you very much.